Hello viewers, this is Shomal Chatterjee and I'm currently pursuing my Masters in Biotechnology from Pondicherry Central University. Today we are going to discuss about a very fundamental chapters in biology. When we start reading, are we able to read from the very first day? I don't think so. Slowly, we develop the habit of reading. First, we learn how to write A, B, C, D, the alphabets. Then we start with a word. Then we learn how to read a sentence. And then slowly we start reading novels and whatever. So we always start with the basic, simple, fundamental things. Cell, the unit of life, is one such chapter. In biology, you will be learning about molecular biology, genetics, microbiology, biotechnology. You will be learning about human physiology, plant physiology. Without cell, the unit of life, you cannot move forward. So this chapter, cell, the unit of life, is that important in context of biology. So without wasting much of our time, let us dive. First of all, what is a cell? Now, you all know from the very first class of biology, we know that there is a cliche line which is said that cell is the structural and functional unit of life. Now, what is the meaning of it? And in our books and in many other places, there are examples. For example, uh, teachers might have said you that when we built a house, right? When we built a house, there we have bricks. The bricks, they are the smallest unit of a building. Without the bricks, you cannot make a building, right? So cell is just like that. There is a small cell. Now, when similar cells, just think of it as in a classroom. In a classroom, we have students, right? Now, maybe your vibe is matching with another person. So you two become friends. Then you two go and make friends with lot of other people, so you make a group. Just like that, there are different type of cells, right? Now certain cells, they have a similar functions, so they will come together and they will form a tissue. These tissues will togetherly form a organ. They will form an organ. The organs will work in unison with each other. They will work together and they will make an organ system. Different organ systems, for example, we have digestive system, we have circulatory system, we have respiratory system, we have cardiovascular system. Movement and locomotion is also done by the skeletal system. So different systems will come together and they will make up an organism. That is the basic. Everything starts with a cell. Everything starts with a cell. In fact, if we think of a human being, it starts with a single cell called as zygote. So cell is that important. It is the basic structural and functional unit of life, meaning it is the smallest living unit that can carry out all the life processes such as nutrition, growth, metabolism, response, and reproduction. In NCERT, there is a very famous line called, it is the smallest unit that can exist independently. Cell, it is the smallest unit that can exist independently. So, if we see this, it is the smallest unit. I hope you can see this. Smallest unit. This is the smallest unit that can exist independently. That exist independently. It is the smallest unit which can exist independently. It is the smallest unit which can exist independently. I hope you can say this. It is the smallest unit which can exist independently. It is that important. Anything less than a cell cannot exist independently. Now, here we have a picture. Here we have the animal cell, and here we have the plant cell. What is the basic difference? What is the difference which is very much visible? Plant cell, we know this is the plant cell. So, plant cell, it is having a cell wall. So, this one is actually the cell wall. But animal cell, it is devoid of a cell wall. Animal cell, they do not have a cell wall. Plant cell, they have a cell wall. That is the basic difference. One more difference, it is very much visible. That is vacuole. If you see the vacuole, this is the vacuole. In case of an animal cell, the vacuole is very small and it is not located in the center. And here, if we see in case of a plant cell, there is a large vacuole. A very large one is present, right? So these two differences are very much visible. There are many other that we will see. Now coming to the historical background. Everything that we do, everything they have a historical background. What about cell? Cell will also have a historical background. This discovery of cell, it dates back to 17th century. In 1665, somebody called as Robert Hooke, you can see this gentleman, Robert Hooke, he saw thin slices of cork under a microscope which he made. That was a very preliminary microscope. So he made a preliminary microscope and just out of curiosity, he observed a thin slice of cork and he was able to see some small blocks. He called them cell. But whatever he saw under the microscope, it was actually a dead cell. The first living cell or live cell, it was observed 9 years after Robert Hooke by someone called as Anton von Leeuwenhoek or Antony von Leeuwenhoek. He was a Dutch microbiologist. He was the first person who saw live cell under the microscope. And he was able to see bacteria, he was able to see human sperm, he was able to see protozoans. So he was the one who saw the first live cell, the living cell. Whereas Robert Hooke, whatever he saw, it was not live, it was a dead cell. He saw cork, he saw dead cell, right? Now, coming to the next one, the cell theory. These three gentlemen, Matthias Jacob Schleiden, Theodor Schwann, and Rudolf Virchow, they gave something called as cell theory. What are the components of cell theory? Let us see. Cell theory was proposed during the 19th century by these three gentlemen. So remember the name. These three names are very important. Matthias Schleiden. We have somebody called as Matthias Schleiden. So this is the Matthias Schleiden, number one. Theodor Schwann and Rudolf Virchow. These three gentlemen, they gave the cell theory. What the cell theory states, this is a very important question. If you are giving exam for 11 boards or for NEET also, sometimes question comes from this area. So this area is very important. Cell theory was proposed during the 19th century and it states the following. Number one, all living organisms are made up of cell. All the living organisms, they are made up of cells. Okay, all the living organisms, they are made up of cells. Okay, so that is the first thing. All the living organisms are made up of cell. If you see the human beings around you, your friends, okay, the animals, the plants, whatever living organisms you can see, whatever living organisms you know of, all of them are made up of cells. The cell is the basic unit of structure and function in an organism. In the first definition slide itself, I mentioned that this is a cliche line. It might sound very familiar because you might have heard it many a times that it is the basic unit of structure and function. If we look into the structure, the basic thing that you can think of is a cell. Anything less than a cell cannot exist independently. Function wise also, the cell, it can carry out all the life processes which is required for an organism to survive. All the cell arise from a pre-existing cell. All the cell, it arise from a pre-existing cell. Cell, it cannot come just like that. There must be a lineage. For example, we have our parents. Our parents, they have their parents. So it is coming like a hereditary tree. It's like a tree, right? So the cells also, they come from pre-existing cell. Okay. All the cell contain the genetic material passed to the daughter cell. As I said, it's just like heredity. From one cell, another cell will come. Okay. From that cell, another cell will come. So it's just like a passing on of the genetic material. Now, what is the genetic material? There is a chapter in class 12 that you will absolutely enjoy, that is the molecular basis of inheritance. Over there, in the first half, you will see that there was a confusion. There was a confusion among the scientists. There was a, what to say, debate that which is the genetic material, whether it is protein, which is the genetic material, or it is the DNA. Actually, there was a confusion in the nucleic acid itself. We know that there are two different type of nucleic acids that we know, the DNA and RNA. So which is the genetic material, whether it is DNA or it is RNA, or whether it is protein which is the genetic material? So there was a confusion. Cells contain genetic material, and now it is very clear that most of the living organisms, not all, because there are certain viruses, they have RNA as the genetic material. So most of the living organisms, for example, if we talk about human beings, we have DNA as the genetic material. This genetic material, it is passed from one cell to another. See, cells contain genetic material, which is the DNA. Now we know it. Passed to the daughter cells. Energy flow, that is metabolism, occurs within the cell. And this metabolism is very, very vital. Okay, in the first chapter of class 11, the living world, over there was a question. Okay, that what is the defining feature of a living organism? What makes a living organism different from a non-living organism? And it was this metabolism. Energy flow or metabolism, it occurs within the cell, and this is something which differentiates. This is one of the defining feature of a living organism, the metabolism, and it occurs within the cell, right? Now coming to unicellular and multicellular. Unicellular, from the name only, it is very clear that it consists of a single cell. What are the examples? See, here we have something called as bacteria, right? Here we have something called as bacteria. So here we have bacteria. Bacteria is one of the unicellular organism. Then we have paramecium. We have amoeba. Amoeba, they can change their shape. And we have chlamydomonas. All of them are unicellular organisms. They consist of a single cell. So we have a single cell. They consist of a single cell. Here we have multicellular organisms, consist of many cells working together. Many cells, they are working together. This is a multicellular organism. And multicellular organisms are usually complex, right? They are usually complex. Then we have this petromyzon. All of them are multicellular organisms. The human, the plants, all of us, we are multicellular organisms. Multicellular, whenever you hear of this term multicellular, you know that multicellular means what? Many cells are there. So if there are many cells, there will be grouping, and this grouping will be actually done on the basis of their function. If suppose there are 100 different cells, now few cells are performing or they are related, they are doing something which are related to digestion. So they will be grouped together to form a tissue. This tissue will further develop into an organ. As I said, these organs will come together and form a system which is called as a organ system. And different organ systems, when they work together in unison, that is what makes up an organism. These are the different level of classifications that you must know. So the basic difference between a unicellular and a multicellular organism is what? Unicellular organisms consist of a single cell, for example, bacteria, amoeba, and multicellular organisms consist of many cells which are working together. Example, humans and plants, right? Now, this is something which is asked. It will not be uh, asked as a direct question in NEET, but in case of your boards, especially in class 11, you can expect this difference can directly come. So let us see the difference between a prokaryotic and eukaryotic cell. First of all, let us see what is the meaning of this prokaryotic. What is pro? Pro means primitive. The word pro, it means what? It means primitive. P-R-I, primitive. Primitive. Okay. Pro means primitive. And karyon, this karyon word, it means nucleus. The karyon word, it actually means nucleus, right? Primitive and nucleus. Now coming to eukaryotic. What about you? Eukaryotic. You means true. You means true, true. Karyon, as I said, it means nucleus. So prokaryotic means primitive nucleus, and eukaryotic, you means true. So true nucleus. That's all. Now let us see prokaryotic cell and eukaryotic cell. What is the difference? Prokaryotic cell, they have a well-defined nucleus is absent. In case of a prokaryotic cell, well-defined nucleus is absent. Their nucleus, it is basically the nucleoid region. No membrane-bound organelles are present. There are so many membrane-bound organelles. Mitochondria is one of them. Chloroplast is one of them. Okay, so there are many membrane-bound organelles, and these membrane-bound organelles are present in case of eukaryotic cell, not in case of prokaryotic cell. DNA is circular and free. Prokaryote example will be what? Bacteria. In case of bacteria, we get to say circular DNA, right? In case of bacteria, we have circular DNA, and this DNA, it is free. And one more very important feature is 70S ribosomes. In case of prokaryotic cell, we have 70S ribosome, whereas in case of eukaryotic cell, we have 80S ribosome. We have 80S ribosome. Then here, in case of prokaryotic cell, we have circular and free DNA. Here we have a linear, and it is nicely packed in the chromosome. Here, as we said, that there are no membrane-bound organelles. Here, we have a membrane-bound organelle. Well-defined nucleus is present in case of eukaryotic cell, but here, the well-defined nucleus is not present, right? Example of eukaryotic cell will be plant, animal, fungi. All these are eukaryotic cell. Now, let me discuss one thing. In eukaryotic cell, in prokaryotic cell, we know that there are only 70S ribosome. But in case of eukaryotic cell, there are 80S ribosome as well as the 70S ribosome. Why? Because 80S ribosome, it is present in the cytoplasm, and this 70S ribosome, see in the bracket it is written in the organelles. Can you name those organelles where 70S ribosomes are present? 70S ribosome, it is actually present in mitochondria and in chloroplast. We know that there is a theory, and the theory is called as what is the name of the theory? Endosymbiotic theory. Endosymbiotic theory. Right? There is a theory called as endosymbiotic theory. Now, this theory suggests that, see, mitochondria and chloroplast, it is believed that they long back they were just like bacteria. They were prokaryotic organelle. They were living independently. Then, because of this endosymbiosis, they were integrated. Okay, they were integrated within a eukaryotic cell. That's why if you see mitochondria and chloroplast, they have their own set of DNA. They can replicate on their own. They are not dependent on nucleus. They are not dependent on the nucleus for their replication. They can replicate on their own. But a major portion of the proteins which are required for their replication, it comes from the nucleus. So in that way, they are dependent on the nucleus. Otherwise, they can replicate on their own. They have their own set of DNA. They have their own ribosome. And these mitochondria, the chloroplast, they have 70S ribosome. Whereas the other portions of the eukaryotic cell, especially the cytoplasm, in the cytoplasm, we get to see 80S ribosome, not 70S, right? Now coming to a cell structure, the eukaryotic cell, let us have an overview. So this is the mitochondria. let me take this black color. Okay, so this is the mitochondria, right? This mitochondria, it will have 70S ribosome. Remember, this mitochondria will have a 70S ribosome. Then we have the peroxisome. We have the lysosome. Lysosome, it is also called as a suicidal bag. We will learn about lysosome in the later slides. This lysosome, they contain what? Hydrolytic enzyme. Then we have the centrosome. We have the cytoskeleton. Cytoskeleton, from the name only, it is very clear, skeleton. So it will give the cell a shape. It will give a mechanical support to the cell, and it will also help the cell to move. It will help the different parts of the cell to move, and it also plays a very vital role during cell division, right? Then we have rough endoplasmic reticulum. In short, we call as what? RER. This rough endoplasmic reticulum, in short, we call as RER. Why it is called as rough endoplasmic reticulum? Why it is rough? Because when we see them under the microscope, since this rough endoplasmic reticulum, they have ribosomes sitting on top of them. So when we see under the microscope, they appear rough. That is the reason they are called as rough endoplasmic reticulum. Then we have this ribosome. This ribosome will be 80S ribosome. These are what? These are cytoplasmic ribosomes. So they will be 80S. Okay. But this mitochondria, they will contain 70S ribosome. Very, very important. So within the eukaryotic cell, we have two different type of ribosome, the 70S ribosome and the 80S ribosome. Coming to the smooth endoplasmic reticulum, ACR, in short we call as ACR. Smooth endoplasmic reticulum, if you see, they do not have ribosome on top of them. That's why they appear very smooth. They are actually responsible for the lipid synthesis, and this rough endoplasmic reticulum, they are responsible for the protein synthesis. Golgi apparatus. Golgi apparatus is also called as the post office of the cell, because see, from here the proteins will be synthesized, from here the lipids will be synthesized, and then they will come to this Golgi. They will come to this Golgi apparatus, right? And in the Golgi apparatus, what will happen? In the Golgi apparatus, it's just like a factory. It's like a factory. The proteins will come. The proteins will be modified over here. Whatever proteins are coming, they are very primitive. Okay, they are very primitive. They are not ready for the industry. It's like that. So the proteins will come here in the Golgi apparatus. The proteins will be modified. The proteins will be packaged, and then the proteins will be transported. The proteins will be transported. Why we have to transport the proteins? Because some proteins are meant for the cytoplasm. Some proteins will be restricted to the cytoplasm. There are some proteins which will go to the membrane, and there are some proteins which are meant for specific organelles. So the proteins will come here, the nascent proteins, the immature proteins will come here. There will be maturation, modifications will be here. Then there will be proper packaging. Okay, then it will be transported. It will be transported. How it will be transported? There are certain signals. Based on those signals, the proteins will be transported. So if it is meant for the membrane, it will go here, not outside. I actually made it outside, so it will be here only. If it is meant for the organelles, then suppose it is meant for mitochondria, so it will go here. Suppose the proteins are meant for lysosomes, so it will go here. And this happens with a lot of accuracy. You cannot make a mistake, right? And here, from here, again, you will go, suppose it was meant for the endoplasmic reticulum, rough endoplasmic reticulum, so it will go here. And if it was meant for the cytoplasm, it will go here only, it will be there. In case of post office, or for example, you order something in the Flipkart, right? Now, sometimes due to manual error, maybe your order, it went somewhere else, and someone else's order, it came to you. This can happen. These are manual errors. But in cell, if this occurs, then the cell will die. It will be a deep danger. So you cannot The cell cannot afford that, right? That mistake the cell cannot afford. You can afford. The Flipkart or Amazon, whatever companies we have, they can afford it. But you cannot afford that, right? Now coming to the plasma membrane. The plasma membrane, let me take a color. Okay. So the plasma membrane, it is made up of phospholipid bilayer. So here we have a phospholipid bilayer with proteins and carbohydrates. Why it is called as a phospholipid bilayer? See these, can you see this? These are phospholipid bilayer. Okay, let me take another color. These are phospholipid bilayer. See, these are phospholipid bilayer. These lipids are called as what? Amphipathic. They are called as amphipathic. Why? Because see, these are hydrophobic tail. These are hydrophobic tail, non-polar tail. What is hydrophobic? Let us break the word hydrophobic. Let us break the word hydro. Then we have phobic. Hydro means what? Water. Phobic, it is similar to the word phobia. So hydrophobic, that means they are afraid of water. So these are the these portions are water. Okay. Look at my cursor. These portions are water, right? These portions are water. So hydrophobic, that means they are afraid of water. So they will try to hide themselves from water. They will try to move away from water. So they are inside. The hydrophobic tails, they are inside. The hydrophilic, philic means love, love towards water. They are polar heads. So polar heads are outside. Hydrophobic tails are inside. Hydrophilic heads are outside. So within the same molecule, there is a polar region and there is a non-polar region. This is called as a amphipathic. These kind of molecules are called as amphipathic. So this is the phospholipid bilayer, right? Here we have a globular protein. See the globular protein. Integral membrane protein. Integral means from the name, you can understand they are integrated. Then we have glycoprotein. This glycoprotein, they plays a very important role in cell signaling. Okay, they play a very important role in cell signaling. Uh, if you are interested in biology, uh, maybe in higher classes you will learn about the cell signaling. It's a very interesting process. To make it simple, cell signaling is a way by which the cell communicate. We communicate with our friends, we can talk, but the cells cannot talk, so they have to communicate in their own way. They communicate with the help of a way that is called as a cell signaling. Let me give you a little more detail. So you can see this. This looks like a receptor. This looks like a receptor, right? So maybe a signaling molecule, it is present here. This will come and sit over here. As soon as it will sit over here, there will be a structural modification. And because of this structural modification, this is what? This is a membrane, plasma membrane. As soon as there will be a structural modification, there will be a series of reactions which are occurring inside the cell. This is called as a cascade reaction. And finally, it will go to the nucleus. And there in the nucleus, it will change the transcription, the translation, and finally, the protein which was about to bring about a change, which was about to bring a change, that protein will be either it will be inhibited or it will be overexpressed. So it's a very difficult term. Just for your interest, I'm saying that it is selectively permeable. Now, this is very important. The plasma membrane, it is selectively permeable. Selectively permeable means it will allow only few substances to go in and few substances to come out. For example, there is a event which is going on, maybe Arijit Singh event. Now you know there are so many uh, problems that can occur, security problems and all, because Arijit Singh is a very big star. So you will have to buy tickets, you will be given a pass, you will have to show that pass to the security person, and the person will allow you to go inside. Otherwise, without the pass, you will not be allowed to go inside. So it's just like that. It is selectively permeable. Only those who have bought the pass, those who have the pass, they will be allowed to go inside. Without the pass, you will not be allowed to go inside. Simple. It is described by a fluid mosaic model. It was proposed by Singer and Nicholson. Remember the year, it is 1972. 1972, this was the year. Fluid mosaic model was proposed by Singer and Nicholson. The protein floats in a fluid lipid sea. Remember, it is not at all, this plasma membrane, it is not at all rigid. Okay. Cell wall is a rigid structure, but plasma membrane is not at all rigid. Okay. Remember this. Coming to cytoplasm, it is a jelly-like matrix between the membrane and the nucleus. So it is a jelly-like matrix between the membrane and the nucleus. Whatever metabolism, the glycolysis, and there are so many different type of metabolism, it all occurs in the cytoplasm. A major portion of the cytoplasm is nothing but the water. So it houses a lot of organelles and it is a site of many biochemical reactions. The membrane-bound organelles that you can see, all of them are present here. The liquid portion of the cytoplasm, okay, the liquid portion of the cytoplasm, it is called as what? The liquid portion of the cytoplasm is called as cytosol. It is called as cytosol. The liquid portion of the cytoplasm is called as cytosol. So cytoplasm is a jelly-like material between the membrane and the nucleus. It houses a lot of organelles and site of many biochemical reactions. It helps in the movement of materials within the cell. Okay, and the liquid portion of the cytoplasm is called as what? Cytosol. Is it clear? A cytoplasm is a fluid-like substances that fills up the cell. The cellular organelles and structures are suspended in the cytoplasm. Okay. Coming to nucleus. Nucleus is the master. Just like if we think of ourselves, who is the master of our body? The brain. Okay. Often we see a lot of memes, right? That dil ki sunu ya fir dimag ka, right? So this is a very common meme. But in case of a cell, there is nothing called as a heart. Everything is decided by the master, and nucleus is like a brain of the cell. So whatever things are happening inside a cell, everything is dictated by the master nucleus. The nucleus is the control center. सारे कंट्रोल जो है वो न्यूक्लियस के पास है. ठीक है, वो जो करेगा वही होगा. So it is enclosed by the nuclear envelope with pores. Now this nuclear envelope is a concept which is only for the eukaryotic cell. Prokaryote के पास ये nuclear envelope नहीं है. और ये जो pore है ना, these are called as nuclear pore. It contains chromatin. What is chromatin? DNA plus protein. What kind of protein? Do you know what kind of protein? They are talking about actually histone protein. Histone is a basic protein which is made up of basic amino acids like lysine and arginine. They are basic amino acids that condenses into chromosome during cell division. Nucleolus produces ribosomal subunits. Okay, ribosomal RNA कहाँ कहाँ होता है? न्यूक्लियस में, न्यूक्लियोलस में होता है. ठीक है. So it is the control center of the cell. It is enclosed by the nuclear envelope with pores, contain chromatin, DNA plus protein. Protein means histone protein. They are talking about histone protein. So what protein they are talking about? They are talking about histone protein. How many histone proteins are there? 4 different types are there, right? 4 different types of histone protein. H1 is the linker histone. We have H2A, H2B, H3 and H4. Actually there are 5 different types, but this I am not considering because this is a linker. This is a linker histone. H1 is called as the linker histone. H2A, H2B, H3 and H4. 2 H2A, 2 H2B, 2 H3 and 2 H4. They togetherly come together. They come together, and that is what we call as histone octamer. The histone, it is what? It is positively charged. And the DNA, it wraps around the histone. It wraps around the histone. Why? Because DNA, it is negatively charged. Can you tell me why the DNA is negatively charged? Why the DNA it is negatively charged? Why DNA is negatively charged? Why the DNA is negative? Can you say me why the DNA is negative? It is because of the presence of the phosphate group. It is because of the presence of the phosphate group that the DNA is negatively charged, right? So that is all about nucleus, right? Now coming to endoplasmic reticulum, network of interconnected membrane. So we can nicely understand this by looking at this picture. See, they are interconnected membrane. It is a network of interconnected membrane. There are so many interconnected membrane. You can see this. Have you seen Takeshi's Castle? It is a very famous show which when I was small, I used to watch Takeshi's Castle on Pogo. So there was a game, I forgot the name of that particular game. So there were a lot of doors, and participants used to cross that, and finally they reach a particular spot. I forgot the name of that particular game. So this endoplasmic reticulum, it looks exactly like that, right? This endoplasmic reticulum, it looks exactly like that. So here we have the rough endoplasmic reticulum RER, smooth endoplasmic reticulum ACR. Rough endoplasmic reticulum, see why it is rough? See, this is a ribosome sitting on top. Another ribosome, another ribosome. So many ribosomes are sitting over here, right? So this rough endoplasmic reticulum, they have ribosome. That's why when you see them under the microscope, they appear rough. They synthesizes and transport proteins. So what is the purpose of rough endoplasmic reticulum? They synthesizes and transport protein. Smooth endoplasmic reticulum, they lack the ribosome. No ribosomes are present. They synthesizes lipids and detoxifies the substances, right? Now coming to Golgi apparatus. Already I mentioned, right? Already I mentioned that what is the purpose of this endoplasmic reticulum? What is the purpose of Golgi? Already I mentioned. In the rough endoplasmic reticulum, from the rough endoplasmic reticulum, we get the protein. But these proteins are immature proteins. So they come or they enter this Golgi apparatus. Golgi apparatus is also called as the post office, remember. So if it is asked in the exam that which organelle is called as the post office of the cell, the answer will be Golgi. So the immature proteins that comes from the endoplasmic reticulum, it enters in this Golgi apparatus through the cis phase. Cis phase is there. So through this receives the vesicles from the ER. So this is the endoplasmic reticulum. See this green color? This one is the endoplasmic reticulum. From here, the immature proteins will enter the cis phase. The immature proteins will enter from the cis phase. Here, in the middle portion, the medial cisternae, in the middle portion, see the series of flattened sacs. These flattened sacs are called as cisternae. What is the purpose of Golgi apparatus? What is the function of Golgi apparatus? It modifies the protein and lipid. Modifies means immature protein या फिर immature lipid वो आ रहा था, अभी उसको मॉडिफाई करना, उसको मच्योर करने का जिम्मेदारी है हमारे गोल्गी apparatus के पास. Packages the materials for secretion or internal use. इधर जो भी मॉडिफिकेशन या फिर मैचुरेशन जो भी होना है वो अंदर होगा. फाइनली वो बाहर आएगा with the help of transphase, sorts and ships the protein to destination. So it's just like if you see a delivery app, for example, let me take the example of Flipkart again. You order something, so they take the order. This is the company, okay? This is the company, or think it as a post office only. So here you are writing a letter and you are giving it to the post office. So this is the post office. Now the post office people, what they will do, they will sort the letters. Okay, they will sort the letters. They will maybe they will keep the letters. This will go to a particular place. Then there are few more letters. It will go to another place. So here the sorting will happen. And finally, according to the address, the letters will be delivered. So the protein is entering through the cis phase and it is going out from the trans phase, right? Coming to lysosome and vacuole. Lysosome, it is also called as the suicide bag. It is also called as the suicide bag. The lysosome, it contains hydrolytic enzymes that digest the macromolecule and worn out organelle. That is why the lysosomes are also called as what? They are also called as suicide bags. The lysosomes are also called as a suicide bags. Very, very important. Lysosomes are also called as a suicide bags. Now, what is hydrolytic enzyme? Hydrolytic enzyme. Hydro means what? Water. Lytic means to break down. So if you're breaking something by using water, that is what is called as a hydrolytic enzymes. The enzyme, what is the work of the enzyme? To catalyze a particular reaction, right? So hydrolytic, if you are breaking down something, the enzymes which are breaking down something by using water, they are called as hydrolytic enzyme. So it digests the macromolecule and the worn out organelles. That is why these lysosomes are also called as suicide bags. Remember. Vacuole storage organ in plant, central vacuole maintain the target pressure. So in case of plant, there is a large central vacuole, and this large central vacuole is responsible for maintaining the target pressure. Okay, but in case of animal, the vacuole size is very small. In case of animal, it is very small. One more important thing, which this vacuole, they have a membrane. See, you can see the membrane. Can you see the line that I'm drawing? This is actually called as the tonoplast. So tonoplast is nothing but the membrane. Okay, tonoplast is nothing but the vacuole membrane. The membrane which is surrounding the vacuole, that is called as a tonoplast. And one more important thing you remember about the vacuole, that is it can act as a storage organ in the plant. There is a large central vacuole, and this vacuole also can maintain the target pressure in case of plants. Remember. Coming to mitochondria. Now, you might have heard of a very famous meme. The meme says that mitochondria is the powerhouse of the cell. It is one of the, even those who are not interested in biology, the engineers and those who do not like biology. Generally, the engineers, they do not like biology because they think that biology is something that you have to mug up without understanding anything. So for them, uh, even if they don't like biology, even if they don't remember anything from the subject, this is a term which they obviously remember. Okay, that is mitochondria is the powerhouse of the cell. So it's a double membrane bound organelle. Double membrane bound organelle. How many membranes are there? Double membrane. So let me change the color again. So this is one and this is another. So this is the outer membrane, this is the inner membrane. So there are how many membranes are there? Two membranes are there. Double membrane bound organelle, and it is also called as the powerhouse of the cell. It is responsible for aerobic respiration and ATP. It is the site. It is the site for aerobic respiration. So there is a mistake over here. I'm very, very sorry for this. So this is a site for aerobic respiration and ATP production. It produces ATP. They have their own DNA and they have their own ribosome. So it is semi-autonomous. Already we discussed about the endosymbiotic theory. Mitochondria, previously it was just like bacteria. It was a prokaryotic uh, cell. It was existing independently, but due to this endosymbiotic theory, it is believed that they slowly integrated inside the cell, and that is why they still have their own DNA, they have their own ribosome. 70s ribosome is there. Okay, they have 70s ribosome. Again, I'm mentioning it so that you do not confuse. They have their own set of DNA, they have their own ribosome, they can replicate on their own. But also remember that a major portion of the protein which is responsible for their replication, which is responsible for their life processes, it comes from the eukaryotic nucleus. So they are semi-autonomous. They are semi-autonomous. This is very important. They are not fully autonomous. They are not fully autonomous. To some extent, they are autonomous, but again, there are some points, there are some aspects where they cannot exercise their full power. They are dependent on the eukaryotic nucleus to some extent. That is why they are not fully autonomous, they are semi-autonomous. So we have inner membrane, we have outer membrane. In between, we have the intermembrane space. See, there are so many porins. Granules are there. These are called as matrix. Okay, these are called as matrix. Mitochondria, they have their own set of DNA. They have their own set of ribosome. Now let me tell you a story, a very interesting story. When you have fever, during fever, you take antibiotics, right? Now these antibiotics, or you take some antimicrobial tablets, whatever it is, these antibiotics, they are meant for destroying the bacteria. They are meant for destroying the uh, prokaryotic cells. After taking the antibiotics, you will observe that there are some courses of antibiotics. Either you take 21 antibiotics or you take 14 antibiotics. So there are certain courses. The doctors will prescribe that. After taking antibiotics, you will observe there are two common things which comes, right? The fever is gone, but after some time you will have some stomach problem and you will feel very weak. So that time the doctors say that you have more fruits, you have more healthy food, and avoid junk food at this point of time. So what is the reason behind these two symptoms which commonly comes? Everybody might have experienced it, right? So why? What is the reason for this? When we take the antibiotic. So if we write it, let me write it. Antibiotic, right? Antibiotic. Anti means what? Anti means against. Anti means against. Biotic means life. So it is against life. Whatever prokaryotic cells are there inside the body, the antibiotic will eliminate all of them. Irrespective of whether they are good or bad, it will be eliminated. Irrespective of whether they are good or they are bad, it will be eliminated. You might have heard about some bacteria which are present in gut, the gut microbiome we call. Those bacteria or those prokaryotic cells, they help us in digestion. Okay, they are beneficial bacteria, they help us in digestion, so they will be eliminated. As a result, we will have some stomach problem and we feel weak. Why we feel weak? Because mitochondria, which is thought to be a prokaryotic cell which existed independently at some point of time, so those mitochondria, they will also be eliminated. And remembering that line, mitochondria is the powerhouse of the cell, it generates ATP, it is responsible for the energy production. So if mitochondria, they are also eliminated, we are deficient of energy. Energy is not produced, ATP is not produced, we do not have enough energy. So that is why we feel weak. So that is why when we take antibiotics, we feel weak because mitochondria, they are also eliminated. And the prokaryotic cell, all the prokaryotic cell, irrespective of whether they are good or bad, all of them are eliminated. So when the beneficial bacteria present in our gut, if it is eliminated, we experience some stomach problem, right? So it's just a story to make it more interesting. Coming to plastids. Plastids are not present in animal, it is present specifically in the plant. So chloroplast, we know it contains chlorophyll and it is the site of photosynthesis. Chromoplast and leucoplast, they are pigments and they also help in some storage purpose. Plastids, they also have their internal DNA and ribosomes. They are also semi-autonomous, just like mitochondria. So here I have given a lot of types of plastids. For example, we have etioplast, we have chromoplast, we have chloroplast, we have leucoplast, amyloplast, elioplast, and proteinoplast. All of them, they have their specific functions. Let me read out, nothing to explain. Etioplast, crystalline prolamellar bodies, precursor of chlorophyll. Etioplast, they are precursor of chlorophyll. Chromoplast, they are pigment synthesis and storage. They are responsible for it. Chloroplast, very famous, photosynthesis, lipid synthesis. Leucoplast, synthesis of fatty acids and amino acids. Then we have amyloplast, storage of starch and sugars. Elioplast, we have storage of lipids and oils. Proteinoplast, sites of enzymes or protein activity. So I've read it out, nothing to explain. Coming to cytoskeleton. So it's a network of protein filament. We have microfilament, we have microtubule, we have intermediate filaments that provide shape, that provide movement, that provide transport, and it also helps in division. So again, from the name it is very clear, skeleton. So it will give us a proper shape, it will give the cell a proper shape, it will give the cell a mechanical support, it will help the cell to move, and whatever is present in that inside the cell, it will be able to move because of the cytoskeleton. And we have then division support, it also helps in division. Cell wall, plants and fungi. I mentioned that cell membrane, it is not at all rigid, but cell wall, which is present in plant and animal, which is present in plant and fungi, it is absent in animal. Cell wall, it is a rigid, non-living layer which is present outside the plasma membrane, but not in case of animal, only in case of plant and fungi. It provides a shape, it provides protection, it provides support, most importantly, it provides rigidity. In plants, it is mainly composed of what? It is composed of cellulose, plasmodesmata, it connects the adjacent cell. So if we look into the structure, see this is a cell, right? This is a cell. This one is the middle lamella. Here we have the secondary wall and this is the primary wall. So it is mainly composed of cellulose in case of fungi. So again, another question that I'm just throwing up. See, plant, in case of plant, the cell wall is made up of what? The cell wall is made up of cellulose. It is made up of cellulose, right? In case of fungi, it is made up of what? It is made up of chitin. It is made up of chitin. The cell wall of fungi is made up of chitin and the cell wall of plant is made up of cellulose. That's all. Transport across membrane. We have two different type of transport. Okay, we have two different type of transport. We have passive transport, we have active transport. Passive transport, again go by the name. Passive transport means no energy is required. For example, diffusion. Example is osmosis. Example of passive transport is what? Diffusion. Example of passive transport is osmosis. What is diffusion? What is diffusion? What is the definition of diffusion? The movement of a molecule from the region of higher concentration to the region of lower concentration. The movement of molecule from the region of higher concentration to the region of lower concentration. So you are moving along the gradient. You do not have to do any work. You are moving along the gradient. Okay. So the movement of molecule from the region of higher concentration to the region of lower concentration, the molecule is moving along the gradient. So you do not have to do a work. The movement of the molecule is from higher concentration to lower concentration. What is osmosis? In case of osmosis, it is more specific. The movement of water molecule from the region of higher concentration to the region of lower concentration. But here is a twist, through a semi-permeable membrane. And we know that the plasma membrane or the cell membrane, it is nothing but a semi-permeable membrane. So the movement of water molecule from the region of higher concentration to the region of lower concentration through a semi-permeable membrane is what we call as osmosis. Here we do not need energy. There is no need of ATP. कोई ATP का जरूरत नहीं है. So passive transport is a diffusion, the movement of molecule from the region of higher concentration. See, so much of concentration to the region of lower concentration through a semi-permeable membrane. Okay, now here we have another term called as facilitated diffusion. What is facilitated diffusion? Facilitated means you are facilitating, facility. From that facility term, you can understand what is facilitated diffusion. What is facilitated diffusion? It means that there are certain carrier protein, or there are certain carrier protein, there are certain carrier protein, there are certain channel protein which are helping the transport, which are helping in the transport. Carrier protein, channel protein, you must have heard of this. So when these proteins are helping in the transport, that is what we call as facilitated diffusion. Okay, now coming to active transport, it is occurring against the gradient, just opposite. Here the movement is occurring from the region of lower concentration to the region of higher concentration. So obviously when you're moving from lower to higher concentration, you need more energy, you need ATP, you need energy in the form of ATP. See, when you are moving from a lower concentration to a higher concentration, you need ATP. Let me give you an example. If you remember this example, you will never forget the difference between passive transport and active transport. See, passive transport, it means that you are moving from the region of higher concentration to the region of lower concentration. So you are moving along the gradient. You do not need any sort of energy. You do not need ATP, right? Suppose you are a very good student. You always come first in your class. Now you are not giving effort. You absolutely stopped studying because you want to fail. So you are not giving any sort of effort. Naturally, the exam comes, you are not able to write anything and you fail. So that is passive transport. You are not giving any effort because you are moving along the gradient. You are moving from a higher stature to a lower stature. So for that, you do not have to give any effort. You fail. But in active transport, if you are a failure, if you are considered a failure, if you are a very bad student, you can always work hard. You have to work double hard to be the first, to be the topper. So you put a lot of effort, you put in a lot of energy, and finally you are now a topper, right? So that is the difference. So in passive transport, you are moving from higher to lower concentration along the gradient, so you do not need any energy. But in active transport, you are moving from lower to higher, so you have to give energy in the form of ATP. What is ATP? Adenosine triphosphate, right? Now let us go through some interesting facts. There are trillions of cells in our body and millions of them die every second, but we feel very normal. Why? Because there is a repair mechanism. The worn out, the old cells are constantly replaced by new cells, right? What is the smallest cell? What is the smallest cell? This is also a very important question from NEET point of view. Mycoplasma. They do not have a cell wall also. Okay, mycoplasma, they are the smallest cell and they do not have a cell wall. The largest cell is the ostrich egg. The longest living cell is the nerve cell. They can live for the entire life, and the cells like the skin cell and the RBC, not RCB, it is RBC, red blood cell, they live only up to few days or months, right? Remember RBC, that is the red blood cell, they lack nucleus and mitochondria. When we were studying about the common features of the cell, we told that these cells, they always have a nucleus because nucleus is the control center, it is the master. So RBC, they lack nucleus, they lack mitochondria. Why? They are against the rule. They said, no, we will not follow the rule. We will not have nucleus. We will not have mitochondria. Why? Because these RBCs are responsible for carrying the oxygen. They have something called as hemoglobin. Okay, they have an iron containing pigment called as hemoglobin, and the oxygen, they bind with hemoglobin, they form oxyhemoglobin, and they carry it to different parts of the body. So these RBCs, they lack nucleus, they lack mitochondria, they break the rule. Why? But still they live for 120 days. Why? Because if they do not have mitochondria, if they do not have nucleus, then a lot of space is available for them. A lot of space is available for the hemoglobin to go and bind with oxygen and form oxyhemoglobin, so that they can carry more amount of oxygen to different parts of the body. And most importantly, if they have nucleus, if they have mitochondria, they will also consume some amount of oxygen. वैसा नहीं होगा. इसीलिए RBCs, they do not have nucleus, they do not have mitochondria. Why? To reduce the oxygen consumption and to increase the space. What is the shape of RBC, by the way? Okay, let me tell you a very important thing about the shape. Different cells, they have different shapes, and this shape entirely depends on the function. RBCs, for example, uh, they have a biconcave shape. Many of the teachers might have told you that you can compare the shape of RBC with that of Hajmola, Sakmola that you have now after lunch, or after the small packets of Sakmola or Hajmola. So it's just like that. The shape of the RBC is just like that, biconcave. Cells, they can commit suicide. This is a statutory warning. Uh, Cells can also commit suicide. This is called as apoptosis, programmed cell death. It is very essential for the development, removing damaged cell and preventing cancer. Okay, so this is a very interesting topic called as apoptosis. Remember, there is a difference between apoptosis and necrosis. Sometimes we interchangeably use these two terms. Necrosis means there was no plan of the cell to die. Suddenly, for example, there is a frostbite. So there is an accidental damage to the cell that cannot be repaired. So the cell dies. It was not a part of the programmed cell death. Apoptosis is a programmed cell death. The cell will try to recover. If it fails to recover, then it will die. That is a programmed cell death. That is a pre-planned suicide. That is called as apoptosis. But necrosis, it's an accidental death. There was no plan for the cell to die. The cell was living happily. But because of some accidental conditions, for example, there is an accident, or there is a frostbite. Frostbite is normally experienced by the mountaineers. So if it is a accidental, that is called as a necrosis. But if it is a pre-planned suicide, programmed cell death, that is called as apoptosis. This apoptosis, do not think that it is bad. It is very essential for the development, proper development, removing damaged cell and preventing cancer. I can give you a very small example. When a child is born, during the developmental stage, when the child is inside the womb of the mother, the I don't know whether you can see my hand or not. The child's forelimb and the hind limb, they are closed like this, okay? Because in between the fingers, in between the fingers, there are certain tissues which are attaching the fingers. So only if the cells, they die in between the fingers, only if the cells die, then only we are able to separate our fingers. So for the proper development of the finger, in between the cells should die, and these cells actually undergoes apoptosis. Then only we can separate our fingers, okay, both for our hands and for our feet, okay. So it is very important. Mitochondria, they have their own circular DNA similar to the bacteria. So it supports endosymbiotic theory, and it is inherited only from mother. Very, very important. Mitochondria, it is only inherited from the mother. Okay. Mitochondria, it is only inherited from the mother. Very important. All the somatic cells, they have the same amount of DNA, but the gene expression is different. This is called as a differentiation. Differentiation. What is differentiation? All the somatic cells are same. They have their same. What is somatic? Somatic means body. So all the body cells, they have the same amount of DNA, but their gene expressions are different. This is called as differentiation. The cell membrane, as I said, it is not rigid, but the cell wall is rigid. Some cells can change the shape, like amoeba, and they can form pseudopodia. For example, WBC, white blood cell, they can squeeze through the capillary wall. This process is called as a diapetesis. So these are some interesting fact, but the subject called as cell biology, it is very, very interesting. So if you are interested, you can definitely go. But as for the class 11 and 12 standards, I gave an overview of the entire chapter. Hope you like it. But before I close this session, let me discuss some questions. And I would love to, uh, uh, if you guys answer all these questions in the comment section. And let me know in the comment section that what are the things I can improve in my teaching style. It would be very, very beneficial for someone who is just starting like me.